made this subtitle called Special Applications and Thoughts on Using the Nanotrac Wave and Microtrac Q. These are the three instruments that we offer in the Nanotrac line. One has a built-in cell, and this instrument is called the Wave. It's available with a Teflon or stainless cell. Depends really on the materials. Some inks tend to stain Teflon, even though that stain can occur. It really doesn't interfere with the measurements. In that case, people may prefer the stainless cell, which rinses purely and cleanly. This is a Peltier temperature control capability for temperature control studies and instability studies. And it has a flow cell for doing titrations. You can also use it for cell rinsing or sample additions. The second model is what we call the NANO-flex. It has an external probe, and you can use any size vessel with this probe. It merely operates pretty much like a pH electrode, a single probe pH meter, and this can just be inserted into or immersed into a sample, and the particle size measurement can be made. Thus, you can use small and large sample volumes. The third is what we call the NanoWave Q, and the Q is a number for a cuvette cell. With this instrument, a set zero is not needed, and there are 4 variable sample volume cuvettes that are available. This has some other uses that I'm going to describe in just a couple of minutes. The technology built into the cuvette cell offers itself to process control conditions and monitoring. I just want to review very quickly, because it's been a couple of months since Dr. Freud made his presentation. I'm just looking here at a dip immersion probe design, and this would be the NANO-flex. And the probe design has a sapphire tip, and this leads to reference beading with the scatter light from the particles. The purpose of this beading with the actual laser beam, which becomes mixed and combined with the scattered light, offers us very, very, very high signals to make calculations. The very first thing that you would see is that the optical fiber carries the laser. It reaches the sapphire tip, and from that point, a reflection will occur, and part of the location right here at the surface, this interface between the fluid and the sapphire tip, allows for reflection. This is the part that will beat or mix with the scattered light. The rest of the laser passes into the sample, and the sample undergoes scattering, and then the scattered light is captured by that sapphire tip in the probe, and the frequency shifted light that occurs when the light interacts with the particle is mixed with the reflection, and it travels back to a detector system for calculations. The cuvette design is much similar, except it doesn't have a sapphire tip. And rather than having the reflection occurring right here at the interface between the sapphire tip and the fluid, it occurs, the focusing point is on the interior of the cuvette cell wall itself, and a reflection occurs here. This reflection really isn't much different than what you would find off of a window as the sunlight is coming in. There's always some type of reflection, a certain amount of light that is actually reflected back, even though the window is transparent. And we use that feature as part of the design in the instrument, and it also affords us some special opportunities, particularly when we start talking about the Debye calculation of molecular weight, and in particular the DNDC value. Now, the first thing I want to mention to you is that there are some ways of using the instrument that have certain applications and capability. Often there's a need for quick means to obtain information on the concentration of particulate in a suspension. And the particle size and the concentration can be determined simultaneously by using what we call the loading index value. The loading index is the sum of all the light, of all the signals that occur when light is scattered by the particles. And in our software, you can actually see all these different signals that are used by the instrument to go forward into calculations to provide particle size. But a summation of these various signals provides us with a linear calculation that varies with concentration. And the nice thing about this is that it affords us the opportunity to determine concentration of particulate. So with each measurement, we provide the size, and that's located over here in these percentiles, and there are a number of other portions of this display that has particle size and a number of different calculations. But also the important part about this part of the discussion is that it provides us with the loading index. And this is the value that responds linearly with concentration. This is a sample of clear coat paint, and this is the loading index value that is obtained by running various volume concentrations of the clear coat paint. And you can see we have a linear response over the entire range, up to about 35% or so. So the particle size can be measured, but also the concentration of pigment or anything else that might be in any type of a paint can be determined by developing a curve like this, which can be done very easily in Excel, and using the loading index value. The thing is, over here, what I'm showing you is that this can also be performed by on particle systems that are bimodal in nature. This is showing a mixture of 50 nanometer particles with 595 nanometer particles. And you can see here's the known amount of the 50 nanometer particles in terms of percent that's being added to the 595 nanometer particles. And you can see, number one, there's a linear response as the amount of 50 nanometer particles are being added to the 595 nanometer particles. So you can do the same type of a thing regardless of whether the distribution is monomodal or bimodal or has a number of modes associated with the particle size distribution. So the linearity really provides us, the linearity of the loading index value provides a means to determine concentrations without going through a lot of drying and other types of gravimetric and other type of quantitative measurements. The next thing I want to mention to you is we have a calculation involved in our instruments that provides for an advanced high-resolution, high-sensitivity accuracy calculation. The high accuracy we're talking about has to do with the high resolution, has to do with peak separation. And this is a mixture of 37 and 97 nanometer polystyrene. And you can see how well those peaks are very, very well separated. Another aspect to this calculation is the high sensitivity. And this high sensitivity gives us the opportunity to obtain a very accurate calculation of the volume percent that's associated with each one of these peaks. And then also the third part of this is the high accuracy, in that we can obtain the size and the width. And I will show you some data in just a second that relates to this particular graph, where we show the size and the width of these distributions as well. Now, in this case, we had a size ratio of 3 to 1. So what is meant by this is that we have 3 parts out of 4 is one size, and 1 part out of 4 is another size. So the mixture is 25% 97 nanometers, and the other material is in the volume concentration of 75%, and that size is 30 nanometers. When we measured this, and this is coming off this graph here, you'll see the measured amount is 23%. The size is coming in at 94 nanometers, and similarly, the 32 nanometers is being computed as being 77% of the volume distribution. All this information is located up here under this mode summary, and you can see we've identified 2 peaks and its related concentration and the width of the distributions. And these are a couple of values over here that are related to another type of calculation, which I'm really not going to go into at this point. But the thing is, we really want to compare these values, and you can see how well the discrimination between these 2 different sizes, the computation of the size, how accurate that's being done, and also how accurately the percentage, the volume percentage, is being represented. Now, to do this, it requires some very special calculations, and this is an advanced calculation that we've recently introduced. And anyone that has our instruments, if you'll please contact us, we'll make sure that if you don't have this capability and you desire it, just please let us know, and we'll make sure that you get a software upgrade. Now, as Dr. Freud mentioned, what we do is not photon correlation spectroscopy, but we actually go through what's called a frequency analysis, and that is necessary to pull this kind of information out of the world of the dynamic light scattering. Cumulants and negative least squares, inversions, it's really kind of ill conditioned to be able to really achieve this type of resolution, sensitivity, and accuracy. The next thing I want to like to talk about is the Debye plot, and dynamic light scattering instruments in general can perform this, and it's something that you do have to use great care with because of the concentrations have to be well defined and have to be prepared very, very well so that errors are avoided. Now, one of the things that we have in the NanoTrack wave is the capability to compute the DNDC value. And if you look over here, this is the molar mass, and the molar mass is, in a general equation, is related to the Rayleigh ratio, which is a ratio of the scattered light to the incident light. And we can obtain the scattered light, as I've shown in one of the previous plots with all the signals. The incident light is just the laser beam itself. But we're also associated with this calculation of the molar mass is a value K. And you'll notice inside this K value is something very, very special, and it is this DNDC value. Now, you'll notice that this K has a very strong influence on the calculation of the molar mass. But as such, it is a square function. So any errors that are associated with this DNDC value the error will also be squared. So correct and careful determination of this DNDC value is very important. Now, with the Microtrac software, we can get the hydrodynamic molar mass as well as the Debye molar mass, and that is done all at the same time. We can use high concentrations to minimize the low concentration error. This is very important, this aspect here. Regardless of the method that you use to determine the DNDC value, high concentrations allow you to minimize the type of error. Now, this DNDC value is an expression to take into account that as particularly smaller molecules change in their concentration, they will have a change in the refractive index as well. So this is why this value is associated with the calculation of molecular mass, is that the Debye plot itself can vary based upon the DNDC value. So again, this is a square function, so errors associated with the DNDC fall right into any error associated with the molar mass. This also can be done with either the probe or the cuvette models. Now, over here down on the left-hand side of the corner, lower left corner, are a couple of things I want to point out. This shows the Debye plot here. And if we look up here, this is the basic equation, and this is an equation for a straight line. And you'll notice here's our Y value over here. Over here we have the weight concentration, which is C. And then we have the inverted 1 over molecular mass. And you make measurements at a few different concentrations, and then the instrument linearizes this through a regression analysis to obtain this value for Kc over R. And then from that, and knowing the concentration, we can calculate the molar mass. Now, at the same time of determining the molar mass by the Debye, here's the value, DNDC, that's been used for this computation. If you look further over here, down here in the left hand corner, the lower left-hand corner, there are a couple things that predominate. One of them is that you can enter the value for the DNDC if it's known, but also you can go forward and actually measure the DNDC value. So there's a little button right here. I'm not sure. The reason why I have this labeled is that because right now it's kind of grayed out, and you would activate the computation of the DNDC value by starting that, and then that would take you to another part of the program so that you can enter information that leads us to the DNDC value. Now, the hydrodynamic molar mass is also presented in the table based upon various concentrations, and I'm highlighting that right here under this column. And that hydrodynamic molar mass is calculated from the particle size. This is Avogadro's number. Here's the diameter divided by 2, which gives us the radius. That value is cubed, and then multiplying that times the density will provide a calculation of the molecular weight. Now, going along with the DNDC value, you notice I've highlighted that, again, this is a square value. And the scattered intensity, or the intensity of the scattered light, is proportional to that value squared times molecular mass times concentration. Now again, because of this ratio is being squared, and we carry that forward into the molecular weight calculation, errors here, again, will undergo a square function. So if the value is known from the literature, then certainly use that value. You can input that value into the software. But there's also a paper, a publication in Biophysical Journal that has this reference here, in which they evaluated over 62,000 proteins for values of the DNDC, and the average value they came up with was 0.1899 mil per gram. And if you would like to read that paper, it's available on the web, and you can evaluate the use of this value for yourself. But it is a handy value if you can't determine it, or if you don't have the value for a particular protein or other substance that exhibits a DNDC. Then you can use what we call the auto DNDC computation and measurement in our Microtrac. Now, the reflected power that I mentioned before, this is the reflection that comes off the interface between the laser just starting to exit the probe and becomes involved with the fluid that's on the other side of the probe tip or the interior of a cuvette. This is obtained as a part of all measurements. Now, this reflected power is due to a difference in the refractive index between the two substances. And the two substances would be the tip of the probe or the inside wall of the cell and the fluid. So as the refractive index becomes greater, then the reflected power would become greater. So increasing or using a fluid of a higher refractive index causes the reflected power to increase as well. And we take advantage of that to compute the DNDC value. Now over here, the dN is really just the delta N, and from this we obtain the difference in the refractive index. And that is from the difference obtained by looking at the reflected power of the sample and then subtracting the reflected power of the water. And these values are built into our instrument and obtained at all times. Now, this reflected power of water, that could be a buffer or some other substance. It really doesn't matter, just as long as that interface is presented to the laser. The second part of obtaining that ratio of DN-DC is the DC can be computed, and again, it's a delta C. Excuse me. The difference in the concentration of the sample And here we would have the concentration of the sample minus the concentration of water. So if we just step back over here to the left, now you can see that we have an expression that describes how the refractive index is changing as the concentration of the material is changing. Now with the auto DNDC, it can only be done with 180-degree backscatter because you need to have these reflections to obtain the information, just to obtain the change in the refractive index. Now each one of these materials, each measurement, once the DNDC is completed, it will be stored with the measurement, and you can use it at a later time. But if the protein changes or something like that, obviously, and you know the value for that new protein or other substance, if it's some type of a saccharide or a polysaccharide or something like that, then you can enter that manually and go forward with the measurements. Now keep in mind, only once is the determination of the DNDC required. It's not something that you have to do with every measurement. Okay, but the caution here is that the fluid must remain the same. If you change the fluid into a buffer or a solvent or something of that nature, then the DNDC value likewise will change. So even though you're using different concentrations of the substance, once that DNDC value is determined, then it's going to have the same value, and it's not necessary to constantly recompute that or redetermine it. Now the NanoWave Q, as I mentioned, this is the cuvette device, and as I mentioned earlier, this just gives you a quick overview again of how the instrument works. Here we pass the laser and the reflection occurs on the inside wall of the instrument. Now this is basically a transparent material. It can be glass, various types of clear plastics, things of that nature. So this gives us an opportunity to take this technology and put it online. So in the process, we are basically going to do the same thing. We're going to have to pass the laser down through a probe. A portion of the laser will pass into a window right here, and a portion of it will go back into the probe to be our reflection. And when the laser passes through this window, then we will perform a focusing right here, and this is an automatic focusing, by the way. But right here at this interface, we will get a reflection which passes back through the laser probe. The rest of the laser passes into the sample just as it does up here with the cell. And once the light is scattered, then it passes back into the probe. The scattered light mixes with that reflection to provide us with real nice high signals, and then we can go forward with the computations that we normally do with any of our Microtrac devices. Now the way this would work is that you have a process system, and the flow through the system is something like this. And this is very schematic, by the way. And then to make the measurement and monitor the process, then you would have some type of a bypass pipe. Now again, this is a transparent window, and depending on the situation, this could be some type of a plastic, or it could be some type of glass material. But right here we have a valve, and this could be a manual valve or it could be an automatic valve. It really depends on the particular process. But all you really need is a bypass pipe. And as those particles are diverted through this bypass pipe, once the sample has gone through, then these two valves would be closed, and then the particles will settle into their normal Brownian motion and diffusion, and then the dynamic light scattering phenomena itself will occur, which is going to produce the scattered light, which we can capture as it goes back through this optical window and passes back into the probe. And then the sample valves, the process valves, can then be opened so that more sample can be passed into the system and provide you with near continuous monitoring of changes in the size of the material. Now, this is a very special application, and this is something that we patented a number of years ago. And when I say we, I mean Microtrac. The person that was really involved with this whole situation was Dr. Freud, and also a researcher by the name of Clarissa Jacobs from the University of North Carolina, I think it was. And the whole idea here behind this device, which is really kind of special and unique, is that it allows you to remove unwanted small molecules such as salts, reducing agents, or dyes from larger macromolecules. So if you have a solution of, just say, a high concentration of sodium chloride for instance, from a salting out procedure or something for a protein, then you would put this into a dialysis bag and then immerse that bag into a large volume of water. Under those conditions, the sodium chloride will start to pass through this dialysis bag because of the very, very small pore size. The salt ions, the sodium chloride ions, will pass through the dialysis bag, but the pores are much too small for the proteins or DNA or polysaccharides or something of that nature to pass through. So you're basically isolating these large macromolecules while purifying them and removing the salt. The same thing happens with your kidneys, which is the purpose, one of the major purposes of maintaining hydration and drinking plenty of fluids, is that it allows the kidneys to function properly to eliminate various ions, urea, and other toxins that can build up in the biochemical system and the physiology of your body, and allows for eventual elimination from the animal system. So this isn't anything that's brand new in itself. The dialysis concept, it's been around for many, 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 many years. But the device I'm going to describe to you is very, very unique. Another thing that you can use dialysis for in the biochemical world is for buffer exchanges or even drug binding studies. And once the dialysis bag containing the material is immersed into the large volume of dialysate, the process of salts leaving or actually entering the dialysis bag will continue until equilibrium is reached. But then at that point, and this can take many, many hours, sometimes overnight, a couple of days, then it's necessary to change the dialysate. And again, this could be water to remove ions, or it could be specific ions that you want to transfer into that macromolecule. But over a period of time, you have to use very, very large volumes of these materials, and it does take generally take quite a while. But at the same time, while all this process is going on, if you want to observe or monitor the particle size changes, any type of aggregation, denaturation, which usually causes some type of precipitation of these proteins and macromolecules, then you would have to open that bag, grab a sample, and then you would have to make the measurements, reseal the bag, and go back through continue the process of dialysis. Now, the thing is, what's desirable is that we can do this on a micro scale, save materials, use small amounts of sample. Now, we can do this with the NanoWave Q. Again, we have the Nanotrac laser waveguide. The laser passes down, and right here at the surface of the dialysis membrane, a reflection occurs, just as it does with the NanoWave Q when a plastic or quartz cell is used. And then the scattered light is produced due to the interaction of these particles with the main laser, and then that's carried back to a detector system, develops those signals that we use for the loading index calculation and linearity. But at this point, we're using it to carry information on the size of the proteins. Now, during this time we have this dialysis bag, what we really want to do is provide this to some type of dialysate. And again, this dialysate could be water to remove ions, or it could be to promote ionic changes within or even increases within the dialysis system itself by this dialysis concept. So the dialysis under a pumping system is passed down through basically this is kind of like a coax cable or something. This is passed down through the center of this conduit, and here the dialysate interacts with the dialysis bag itself. But then it continues on its way and passes back out of the system. Now this can be a recirculating system so that we have a process that's kind of moving like this, or it could be a single pass, or you could take this and put this out to waste if that was desirable. But eventually, if this is under a recirculation system, there would be an equilibrium reached where the concentrations of those ions inside the dialysis bag and in the dialysate will reach equilibrium. But all during this process, particle size measurements can be made. And the nice thing is that you're using a very, very small cell, small amount of material to do this measurement rather than having very, very large volumes of material that I discussed up here under the normal dialysis conditions. These larger volumes typically are used for preparation of larger amounts of material. But before you go through all that trouble, the dialysis membrane and this device that Dr. Freud developed provide you with using very, very small amounts of material and very, very small volumes of the material. And if you'd like to look into this a little bit more, it's under the United States patent. Paul's name is right there with a couple of other people, and this is the patent number. So these are the nice features of this approach. It avoids the need to pull sample to make measurements of the size. Uses very, very low volumes and sample amounts, down to the order of about 40 microliters or 0.04 ml, and small amounts of the sample. So this is the process, and you can see that this could afford protein chemists a very nice system, especially in an R&D environment where small amounts of enzymes and various types of proteins might be measured.
Thank you for checking out our on-demand presentation. We hope you enjoyed the content. Our particle analyzers have been trusted by the materials science community since 1974. Let us show you the value our solutions provide. We would be happy to arrange an on-site demo, or you could send samples for complimentary analysis. To schedule the demo or sample analysis, simply email Jason Noga at the address on the screen to get started. For more information on our entire suite of particle characterization solutions, please visit microtrac.com today.